Phase 2 is to confirm that we have efficacy, most importantly on the bottom line. It sets us up for a very important decision, which we call product decision. By then, we should understand what the drug does, that it's safe, that we can go into thousands and thousands of patients, and that we're willing to invest $500 million in doing that. It also should mean that we understand the ideal dose, who should receive the drug, who should not receive the drug, how are we going to position it, so that when we go into phase 3, we have the ideal population. And it sounds easy, but it actually is not. You have to understand where you would put it in the treatment algorithm. Are you going to put it in early? Are you going to put it in late? Is this good for somebody who's already on insulin? How are you going to position it? And then you got phase 3, which is really where you have to prove it. So now we have ideas, you think you know it, now we have to prove it. In phase 3, you have to prove to the regulators and prescribers and get the data and prove that the drug is better than your competitors. You have to prove that it stops diabetes if that's what it does. You have to prove that it does something to lipids, etc. So this is a very heavy lifting where you have to get all the documentation that you can claim to physicians that this drug does so and so. We do that in a very tight connection with the marketing people, because we need to tell the physicians what the drug does that helps. So we also do a number of small boutique studies where we look at additional benefits of the drug. So, and then switching from that to what I think ties into my colleagues here, mode of actions, which relates to targets. There are a number of mode of actions to control glucose, and I've touched on those. Hepatic glucose output, glycogenolysis and gluconeogenesis are important. I think they have been proven quite hard to target and maybe not that safe. Hepatic glucose production directly to target some of those pathways have not been proven easy to do. In the pancreatic islet, clearly insulin secretion is our number one priority, but it has to be glucose dependent, otherwise you develop hypoglycemia. In the muscle, we're talking glucose disposal. That's where the glucose goes together with the liver. And in addition to lipid deposition, which is associated with insulin resistance in the muscle, we also obviously are very interested in the insulin signaling pathways in muscle and mediators that impair the insulin signaling pathway. I think interestingly, the mitochondria actually could be a unifying target, but it's a little bit scary from a pharmaceutical perspective because they are ubiquitously present in all tissues. So those are the things we look for. What we also look for is things that would make us stop the program. If you have weight gain, it's a significant negative. Who wants to gain weight if obesity was your problem getting diabetes? And some drugs do that. You don't want hypoglycemia. It's risky and getting more important after EASD and ADA. Is the liver safety? You want to know whether there are any issues with the target. So if the target is a receptor that is in the islet, but it's also in the pituitary, maybe it's good in the islet, but it's bad in the pituitary. So you want to know where it's expressed. Is it safe to target in all tissues? Off-target effects is just what we cannot predict, but it's whether it hits something else. If the molecule hits your target, but it also hits an important receptor or channel or enzyme, you're in trouble. Drug-drug interaction is commonly screened out by hepatocyte assays, and the last I'll skip. So what does a profile have to look like to be successful if we start to look for a new drug today? Well, you need as a bare minimum glucose lowering, and I set here the bar at 0.7% in HbA1c. Probably it has to be 1% or more, but you want to know that it's sustainable. Ideally, it would cause something else, which is what we call glucose plus. So we say it has to do glucose, but that's not enough. It has to do more than that. So weight loss is one. Preserve insulin secretion. If you can do that, we believe you can stop diabetes. That would be enough, certainly. Does it improve lipids? If it does so, you can come out with an outcomes trial and you can show it actually improves survival. Does it improve blood pressure? Same reasons, complications. But also it has to be additive. If this drug cannot be added to very cheap existing drugs, it doesn't really help. It has to show that the mechanism can be added on to potentially other therapies unless it's really superior. And then dosing, ideally it has to be once a day. Our patients, if we are successful, would have to take the drug for 15 years. It's very inconvenient to take an injection 3 times a day for 15 years if you could take 1 pill a day for 15 years. So that matters. But if it's an injectable, and insulin is, and the GLP analogs are, it's the volume, how frequently do you have to dose, etc. I'm going to switch from here to, I said Lilly is an old company, 1876. We also look to the future. And we have a model that we are transitioning into, which is called Fitnet. Lilly is a fully integrated pharmaceutical company, and we want to get into a fully integrated pharmaceutical network. So where we went from the hub and spoke model, where everything was Indianapolis, I apologize for the slang with Indy, but Indianapolis-based, we're moving away from being Indianapolis-based to really reaching out to new centers where research is going on, like here in Qatar and other places. We have built a center for integrated biology in Singapore, set centers up in India and China and other places. We're trying to break down those walls and engage in a network. And why do we do that? Well, no matter how big we are, that we will have gaps, and we will have other areas where people are better, where there's better expertise, and we shouldn't be doing it if we are third best or fourth best. We should go to the ones who are the best. And that what it means is that if we don't have the core capacity or capability, we should go somewhere else, and we're willing to do that, and we're willing to do less in Indianapolis and do more anywhere else in the world. Not doing it ourselves, but working with others who actually can do it. So we're transforming from this FIPCO into a Fitnet model, and we do that to strengthen our focus and give us more opportunities. And the bottom line, you can see that it's not just, we are not transforming to transform, but it's a way to get us into the future so that we can produce more and get more innovation and get access to more innovation. And then finally, I'll touch on something that I think could be potentially of interest, which is a virtual development, drug development model we have called CORS. CORS is a small unit with only 24 full-time employees. It uses external experts, so it's really a number of people who don't make decisions themselves. They ask everybody else and are reaching out to them, and they do drug development. And the concept really is that instead of the standard model where you have everything in parallel and paying very heftily for it, you do very few things and you do it very quickly, and you do your killer experiment. Does the drug really work? Is it really better? Well, if you've not paid for everything else, it's not going to be faster. The pharma tended to have this success-seeking behavior. You were sure it would work, so we did everything, and it didn't work, and you paid all that money. So we go from that to say, we don't want to get there quickly, we want the truth now. If we know it works, then we're going to invest later. And that's really the concept in this. And within 2.5 years of operation, it's worked on 18 molecules in the clinic, 7 already finished through proof of concept. And the industry standard to get to proof of concept is $30 to $40 million, and they do it for $6 million. Should Qatar consider getting into early stage clinical development, or should you consider doing that? Lilly is more than happy to work with you through our model, and should you engage in that sense, we'd be more than happy to work with you. It sounds very good, but this is actually not enough for us. We need more of those kinds of models. We need more where we develop virtually or develop outside of Lilly, so we're looking to more places to actually do that. And it's a very lean development machine, and it works very well for us. It's not good for every molecule, but it's good for those where you really need your killer experiment very quickly. So, in summary, the need, yes, there's still a big unmet medical need. I don't think we do too much for diabetes. There's really hardly any drugs that shown they do much to diabetes at all in terms of long-term outcomes. The market is very big, and it's growing. The process for drug development for diabetes, it sounded maybe, I don't know if you think it sounded complicated, but it's very, very easy for diabetes. You know very quickly if it works or it doesn't. So diabetes drug development is easy. So don't fear that part of it. The targets, there's a sparsity of targets, and I think the approach taken here looking at genomics is very important. The risks is mainly on the safety. It's not whether it works or doesn't. You typically know that. It's really whether you harm the patient. The future, I think that it will be that we and others will not do things alone. A drug will not be developed in one place. It will be developed by a number of partners who together bring that to market with a shared risk, but also shared reward.